watching live. This session will be a roundtable discussion, and it is entitled Tales of Ideas Gone Big, and it's focus on translational science in continence. Just a few words about what we mean by translational science. Well, most people identify translational science as from bench to bedside, but it's rarely possible to take that as one step, going directly from the bench to the bedside. Rather, it's a number of processes, and usually you divide it into two different sections, which we call T1, and that means that you have an original basic science observations that you, by different tests and steps, bring up to early human trials. And then the next step is, of course, to implement what you get in phase 2 and 3 trials to the clinic and to common clinical use. So we are now trying to give some examples of translation, ongoing translation, why it's not always successful, and how we could maybe improve our way of translating so we actually can enhance what is going on from bench to bedside. The first speaker here will be Laurie Burder, who will talk about the translational impact of preclinical models for LUT dysfunction. Please.
Well, thank you, Karl-Erik. And so as mentioned, I'm going to be talking about the translational impact of preclinical models on lower urinary tract dysfunction. So while a model is clearly not a human, obviously the information provided by these models has really a profound impact on human health. And these are my disclosures. Animal models are crucial for clinical translation. They're necessary to gain an insight into the pathogenesis of disease, to establish any non-clinical proof of concept evidence for any given target, and finally to provide early evidence of a potential safety risk. Any medicine or therapy must be tested in a suitable animal model before clinical trials in humans can occur. But results from using animal models to predict success in clinical disease experiments have been roundly disappointing. Now, there are a huge number of possible reasons why, but obviously diseases are incredibly complex, and animal models do not really adequately model the human condition, which is complex in itself. So this begs the question, is there a need for standardization of a model? Well, the qualification of any animal model implies that a specific animal, given a specific challenge agent, can produce a condition that, at least in multiple aspects, corresponds to a human disease. Now, whether or not we need this standardization is a matter for debate. Studying symptoms can certainly lead to early diagnosis of a chronic disease. We know that changes in symptoms, behavior, or even traits may be regarded as an early sign of a disease process. And so recent studies in animals have shown a link between early changes in olfaction and eventual changes in cognitive decline. So should these studies bear out, this could actually point the way to early diagnosis and even treatment. Another example is ATP. A huge number of preclinical studies have shown that ATP released in heightened amount in visceral systems, notably the lower urinary tract and the airways, can contribute to afferent sensitization. A number of preclinical and, of course, clinical studies have validated the therapeutic promise of ATP antagonism at P2X3 receptors. Both respiratory and urologic disorders are dominated by very similar neural mechanisms. We know that ATP can evoke symptoms, The concentrations are highly elevated in pathology, and of course the airways and the lower urinary tract have very similar traits, both in morphology, pathobiology, as well as pharmacology. We know that the drug discovery and development process is obviously very lengthy and expensive. For every drug that makes it to patients, 10,000 or more other compounds obviously do not. And of several thousands of diseases affecting humans, only a minority have approved treatments, and many have adverse side effects. So to increase the translational relevance, biomedical databases have been used to construct what is called a symptom-based human disease network. This is to examine a potential connection between clinical manifestations and perhaps underlying molecular interactions. And so in summary, there are a lot of challenges. To identify the phenotypic characteristics of patients that are most predictive of treatment outcomes, and the measurement tools or even endpoints that are best suited to evaluate these characteristics. We know that animal models of disease rarely captures all the key aspects of corresponding human disease. And we know that disease progression clearly changes over time, yet there's little to no preclinical evidence that has really looked at this to uncover the rationale for these changes. But finally, many systems do share many similarities between pharmacology, morphology, and certainly pathobiology, so lessons learned from other organ systems certainly can be applied to the lower urinary tract. And I'll stop here. Thank you.
Well, please, we could discuss and direct some questions to you. I mean, animal models have been used and misused, and are well, thinking about using animal models in a way, at least in some occasions, that are, well, let's say, a waste of time, because we know that, as you said, no animal model can mimic human disease. So can you give an example of animal models that actually have been directly, well, finding in animal models that directly has been translated to, well, a new drug? Or do you have to go from animal models to human tissues and so on before you can say with certainty that what you find has any relevance for a clinical development?
No, I think that's a good point. And so I think there are a number of ways to look at it. It depends on the endpoint and the treatment. I think it's not all that common for animal models to be used directly translatable to a human disease or target. For example, P2X3, though, have gone from a number of animal models to a target, both for chronic cough, maybe not so much for OAB or IC, but that has been done. I think it's very necessary to take human tissue to validate potential biomarkers that have been looked at in various animals.
Do you have any comments, Guy, to this? I mean, you work on the clinical side. Can you give an example of that you actually have been using animal data to guide you when you implement well, early clinical trials to clinical praxis?
Yeah, I mean, this is an excellent question. If only it was accurate, we would save so much money and time. But take, for example, for overactive bladder, you have the cyclophosphamide model, and you also have the proximal urethral constriction model. And I think, it's very difficult to know, well, is the cyclophosphamide model the one for interstitial cystitis, or is it for OAB? And when you get to the proximal urethral constriction model, are you talking about OAB? Are you talking about LUT secondary to outflow obstruction? So I think that, as Laurie has articulated very clearly, there is no one model, but you have to try and look at it in different ways. And what you're asking is, what is the model that has shown us the way? I honestly don't know.
Do you know any, Thomas?
Yeah, I'll answer the question like this. There are, different types of innovation, and of course, if you want to go for the radical innovations, you need a lot of data, which may include animal models. But a lot of the innovations, and I'll come to that in my talk, are incremental innovations, and there you can rely on data from other experiments, and you can do a lot with incremental innovations, and you can go directly to the human, and that is where I think we should be to a much larger extent. And if you do innovations in that environment, they are more predictable. You are more likely to come up with a positive outcome as compared to when you're starting early, in the animal scenario. So I think that's sort of a rethink that we should perhaps do.
Yeah. Well, I have another question to you. I mean, do you think that observations or animal models and data from animal models actually have led us in the wrong direction? I mean, let's discuss, for example, you brought up cyclophosphamide-induced cystitis. Of course, we have a cyclophosphamide-induced cystitis in humans. Is that identical with interstitial cystitis? Obviously not.
Not, yes.
And also, when you induce interstitial cystitis, cyclophosphamide-induced cystitis, you either treat the animal systemically, but what is producing the cystitis is the metabolite, the acrolein, that is, working from inside the bladder.
Yes.
I mean, if there is a model of interstitial cystitis, which is the toxic factor in interstitial cystitis that actually induce the disease, we have never found any toxic agent in the urine that induces interstitial cystitis. Are we, the animal data, are they leading us in the wrong direction? What do you say?
I think they provide some evidence of mechanism, but I think in terms of a particular target, anytime you induce injury or inflammation in an otherwise healthy animal, then I think it's a bit difficult to point to a particular target.
Okay.
Or factors.
Yeah.
Eboo, you were...
Karl-Erik, I just want to say that this is meant to be a round table. Unfortunately, everyone's up there, and the microphones are down here. So we're hoping that you guys were going to give us some answers. Please, come up.
Hi, Toby Chai from Boston. Very interesting topic. I think, as a urologist, thinking functionally is a challenge because, surgeons are anatomists by nature. So you're asking us to think mechanistically when obviously mechanisms are very hard to understand. And it just actually, in the area that we deal with, obviously, in voiding function, I've been taught this new concept of a socio-ecologic model of this disorder of lower urinary tract dysfunction, if you want to use that term. So, it's pretty complicated. I don't want to sway one way or the other. I think animal models are obviously very, very valuable, but they'll never be the exact answer because, just the concept of animal voiding, it seems like we keep on saying, well, if we do something, we change voiding, we know what's going on. But I recall the paper that Claude Desjardins wrote from UVA, the science paper in 1973 about studying voiding spot. It's really a social behavior. And, we look at voiding an animal and say, well, that says this animal has overactive bladder when it's obviously a lot more complicated. So I'm probably not addressing anything specifically that Karl-Erik is asking or what you've presented, Lori, but I do think animal models, in my personal opinion, are valuable, but it's so much more complicated. I don't think we should try to answer that question. I don't think that's the goal. I think there's value in the animal, but we just have to put it in perspective of what a clinician sees and how this particular problem of lower urinary tract symptoms, dysfunction, whatever you want to call it, can have a very broad, all the way up to the social behavior of an animal, to something very specific. And I will also then tell you about my encounter with a psychiatry chairman at Yale who very interestingly said, you know, in my younger days, I used to think one protein, one disease. Well, you know, he's a chair of psychiatry, and he goes, it's so much more complicated. And so, you know, he's managing a faculty of sociologists, psychologists, psychiatrists, behavioral interventionists. And I think, you know, thinking more broadly will help us. But anyway, so much for my editorializing. But I believe in animal models. That's the bottom line, despite all that.
Yeah, well, everybody believes in animal models. It's only don't have too great expectations that they can explain everything. So thank you. And now it's my turn. Well, I will give some examples of continence drugs of the future, how, where they are in a translational stage, and how we can expect in the future to see drugs that successively will be implemented, hopefully, into clinical practice. These are the current targets we have. And you all know that these 4 groups, there are Me Too drugs in development, which are slowly implemented into clinical practice. For example, among the beta receptor agonist, Vibegrone is one example that the drug is slowly implemented into clinical practice. And there are others. But let's look forward. Laurie mentioned, for example, ATP and P2X receptors. And of course, antagonists would then be of interest. But there are several other areas that have evoked a lot of interest, particularly concerning their effects in the lower urinary tract. And these are the TRP channels. Much discussed is also the cannabinoid system, and there we have both agonist and antagonist, and also inhibition of the degradation of the cannabinoids that actually, and endogenous cannabinoids that actually can be, well, interesting from many points of view, also in the low urinary tracts. And we have also all this big group of CNS active drugs. Where are we at the moment in translation to get drugs that actually work through CNS mechanism in controlling micturition? And do we have other options? To start with the P2X3 receptor, there are many current antagonists that has been described in the literature. And of course, they seem to be very promising. As you can see here, they, in an animal model of micturition, you can delay the first, before we can increase the threshold volume, and you can decrease the frequency, and also you can you don't have any effect on the amplitude of the contraction. This seems to be an ideal substance. Decrease frequency, increase bladder volume, and don't decrease the contractile strength. And of course, also the mechanism of action is what we thought it would be, a decrease of afferent activity. And this is, for example, when you give one of these antagonists intravesically. So if we are shortly summing up what we have with the P2X3 receptor antagonist, is a very nice clinical rationale. We have some promising, I have a question mark here, because nothing much has happened in the low urinary tract with these antagonists in the last 5 to 6 years. And there are no clinical experiences on lower urinary tract disorders that actually have been published. But still, this is an area where we can expect some translation from the basic science observations up to the clinic. Everybody is expecting trip channels to be involved in almost everything. And in the lower urinary tract, we know that they are involved in mechanosensory transduction and nociception. And there is an abundance of preclinical information that suggests that these drugs could be active or effective both in bladder over and under activity, and in addition to bladder pain. So what is the real evidence for these drugs being, promising drugs for treatment of lower urinary tract disorders? Well, you find these trip channels in the urothelial cells, in the nerves, both in C fibers and in A delta fibers, and you also find them in the trousseau muscle cells. It means that they can affect both urothelial function, nerve function, particularly afferent function, and the contractility of the trousseau. So what do we find? Well, selective antagonist, and the most investigated one is the antagonist for TRPV1. This looks very promising. You can increase the interval between contractions in the rat models. You can also get correspondingly an increased voiding volume. There is no effect of these drugs on contraction, the trousseau contraction, and you can decrease afferent activity. Well, very promising. But there are also, of course, since these drugs are not only, these receptors are not only found in the bladder, there are side effects that actually limit them. For example, TRPV1 antagonist causes hypothermia and the loss of noxious heat sensation, and that can lead to thermal injuries. And this has delayed the clinical trials. But even if we have now a second generation of TRPV1 antagonists, which do not produce hypothermia, and which also are nice concerning the heat sensations, none of them has been tested in LUT disorders. And these drugs have been tested clinically in, for example, pain disorders, but none of them has actually shown a very convincing effect. So to summarize with these, there are very promising data, but we don't know if they do work in human lower urinary tract disorders. The problems with side effects, it's not only hypothermia, it's hypothermia concerning TRPV1, but some other drugs causes M8, for example, causes hypothermia. And maybe there is a potential for further development, but still there is a lot of work before we can say that we have a product that we can try to really test in humans and to get into clinical practice. So what about the cannabinoid system? People are mostly interested in cannabinoids in psychiatry, but there are some very interesting effects of both exocannabinoids, I mean drugs that you synthetic drugs and so on, or the endocannabinoids. We have FAAH inhibitors, which actually decrease the degradation of endocannabinoids and causes some interesting effects in the lower urinary tract. And also, cannabinoid receptor agonists have been found may be useful in, for example, bladder problems in multiple sclerosis. But there are no really good trials going on, even if you have found that FAR inhibitors can depress both A delta and C fiber activity in animal models. We have no real clinical data to support that cannabinoids should be introduced and tested in lower urinary tract disorders. So if we summarize that, there are promising preliminary data for agonist exocannabinoids in MS, if we are positive. Most people say that the trial is too preliminary to allow any real conclusions. Endocannabinoids, the FAR inhibitors, are definitely promising in the animal models. If that really means anything for the clinical situation, we do not know. But again, there is a potential for further development and for translation up to human use. And then we have, of course, the CNS active drugs. There are a lot of them, and to be very short, there are several principles that seem to work, but they have low efficacy and unacceptable side effects. But there is a great potential for further development and to prove that some of these drugs actually can work for effective treatment of lower urinary tract disorders. But still, if we are looking at drugs that are currently under investigation, there have been K-channel openers that have been tested in the clinic with negative results, and still there are new aspects on both BK and SK channels. For example, you can take the BK, the big calcium-dependent potassium channel, the gene, and inject it into the bladder, and you can actually improve overactivity by doing that. The antifibrotic agents have recently opened up a new horizon for drugs, but we have no more than preliminary animal data, and the clinical potential for antifibrotic agents in, for example, BPH and underactivity would be fantastic. And still more fantastic could be gene therapy. There are different ways of using gene therapy, but none of it has actually been translated the full step up to human use. And humanized antibodies, we have had tested, for example, antibodies against NGF with not a great success. So if overall we summarize what we can say about what is going on the translational market, so to say, there are several unexplored targets, many promising animal data, but there has been a very, very slow translation. And at the moment, we have no really new drugs that are ready for immediate clinical use. Thank you. So, do you have any comments?
Well, Karl-Erik, I think you addressed the main problem, that there's lots of information from basic science that would suggest that we have therapeutic targets, but they're not getting funded. And so that's a major issue of why not.
Yeah, well, do you think it's only funding? That the drug industry is not interested in funding many of those very interesting ideas? And don't I see that there is some kind of potential? I mean, we have 4 major drugs dominating the market. Maybe the drug industry, who must do these very expensive trials, do not see the potential that you can take, or you can maybe be as good as what is existing, but you have no real chance of taking over the market or so on. Is that what you think about?
I think
Yeah, yeah, please.
Thank you very much. My name is Svanberg from France. I have a little bit of a Swedish accent, as you might hear. I would like to comment on you, and it was a very nice segue there about the pharmaceutical industry, since I happen to be part of that for the last 20 years. And what is really interesting is these are the targets that you describe and the potential which are in there. And I'm curious, having been to this Congress and several of them before, is the what should we call it? The perception of the diagnosis per se. Because if you have a cancer, you will die. And if you pee in your pants, I mean, who cares? Put on the washing machine. So the risk averseness in developing a gene therapy or a humanized antibody where the side effects could be significant and the treatment effect could be very potential. I think that comes down to benefit risk. This is not a life-threatening condition. I was very happy to hear today that FDA has actually defined incontinence now as a serious condition because of the day-to-day impact on functioning. But what can an organization like ICS do to put the disease on the map as something which is not just, a little bit weird, into actually being a disease that needs treatment? And what does then success mean? Complete continence, of course, but you mostly don't cure cancer, actually, but you still treat it.
Yeah, well...
Curious, this is more like a round table. It's discussion,
of course.
obviously.
You know that we have 4 agents that actually are reasonably active. There must be some incitament for the drug industry economic gain to put the money into studies into any of these categories that I listed. Because, well, they seem to be good, but even if they are good, and even if they work, and there is a positive proof of concept, is it good enough? I mean, could you get a piece of the market that actually...
That's obvious. But the question is, do the people in the industry understand that? And as ICS being the, if you like, the intellectual arm of this entire this therapeutic area, needs to communicate with industry. I think we need more interaction. If academics stay away from actually engaging with the industry to tell them what unmet needs are, the people in the industry who may not have a background in neurology at all won't understand the need. And unless they're persuaded, these are big risks. In my talk, I'll talk about why these are big risks, but they have to be convinced it's worth doing.
I just want to throw in the question that we are forgetting a quite important player in this troika or the triad, which is the health authorities. So FDA acknowledges that this is a serious condition because of the day-to-day impact on functionality. I actually had an email just last week from Health Technology Assessment Group in Europe saying that we are not reviewing XYZ because it is not a debilitating condition.
Mm-hmm. Well,
so, I'm not so sure this is the academic and the industry. I think that there is some significant common ground there, but it's not a life-threatening condition. We can agree on that. It's not even debilitating. So what is it? It's
well, there's a lot of money in incontinence. You go to pharmacies in America and you see aisles upon aisles of diapers. So, I mean, and the thing is, the health authority's job is to reduce costs, right? That's what they want to do, reduce costs. So they're not interested in new medicines. They're going to increase costs. And so, but patients want it. So again, it's advocacy. You need the patients to tell the health authorities, we need this.
No, I agree. Incontinence is really one of the number 1 reasons why people lose their independence in nursing homes, which is a big cost. But also in terms of, looking at various targets, I mean, there have been some very large studies for IBS, for CRF, and so those antagonists work incredibly well in the animal models. And so there were 3 major drug companies that were interested in that study, and they basically failed. They worked on anxiety, but not on motility. So sometimes it's actually trying to, link up the basic science, the preclinical and the clinical work in terms of what should be the proper endpoints so that a drug has a chance of succeeding.
Please.
Hi, Dick Jensen, Netherlands. Karl-Erik, again, great talk and very clear overview of pathophysiology. I have two comments and I would like to hear your views about it. First one about the cannabinoids. I've got a lot of patients that are on with bladder pain syndrome with cannabinoids. They are already on this type of medication, and I don't know if there's any evidence what it does on their urgency complaints, which is of course very much common in these patient groups, which might be also very easy to look at this because they're already using these types of drugs and see what their urgency complaints will affect this type of drug. And second one, I would like to overview because a lot of these pharmacological trials in humans start with systemic drugs. Why don't take the step previously and look at local administration through, and it's more complicated because you have to use catheters and so on, but it does eliminate a lot of the systematic side effects if you would try it like that. So maybe it's sort of a step in between that might say, okay, if the drug is effective, but maybe the systemic effects are worse, and we need to design a little bit more about these drugs.
Yeah, well, that is, of course, what we talk about, bladder selectivity. That is the big problem. You can't hit the bladder because there are so many other things in between. But intravesical administration, either through injection or just instillation, has never become particularly popular, and I think it's a matter of convenience.
Yeah, I think so too, but I think a lot of these drugs are sort of cancelled, which might have a local, if you put it locally, have a good effect, but it's never investigated, and maybe you will lose very valuable therapies for very therapy-resistant patients, and you will never know it because it's never been tried. So I think that's something worthwhile to investigate, especially when there's a lot of these refractory patients that we have here currently.
Yeah, well, tell that to the funding organizations and we will do the studies.
All right. Hey, thank you.
It's you, Thomas? Yep.
Well, thank you very much. I will be very pragmatic. I will address pragmatism, practice rather than theory. Theory is good, but unless we can turn that into practice, it will not be useful for any of our stakeholders, patients, society at large, and so forth. It's good for research, but we need to get it out there. So I'll be very pragmatic, and I'll give you some principles which I use in practical innovation. And then I'll give you a tale as well. First of all, some definitions. So what is research? Well, research is actually a new theoretical concept or discovery. And that theoretical research finding cannot be patented. So if you want to go for an innovation, you have to think differently. So an invention, that's a new composition. It's a manufacturer, new method of manufacture, it's a machine or upgrade of an existing innovation. And that must satisfy three criteria: novelty, inventive step, and industrial applicability. That's what you need to get at if you want to do an innovation and patent that and drive that through the system. So innovation is actually a new solution or invention, creating new values to somebody out there. Product development, commercial development, yeah, that's another process, very tedious. We have to abide to the rules, etc., etc. So translation, well, that's a top-down strategy. But I would argue that there's also a bottom-up translational strategy. So if you go top-down, you try to translate research into innovation and development and ultimately values out there. But if you use a bottom-up sort of a translational process, you translate medical needs out there, the reality, the needs for patients and stakeholders into innovation and development. And that is the process that I'm trying to do, and I'll tell you some of my principles. Very pragmatic, which I use when I select a project, because I want to know that it creates values for patients out there. So I go for, very interestingly, as you pointed out, topical mucosal administration projects. And that is because it's much easier to come to a product if you do that. There's a lot of the admin steps that you need, that you do not need to do, or you can avoid it. And by avoiding that, you avoid a lot of costs. I select indications with a high incidence or prevalence, because if you are going to do something valuable for yourself and for patients out there, you need to have more or less a chronic treatment. And that creates, of course, potential cash flow for the industry that is probably ultimately going to buy your innovation. I use and work with known substances because the regulatory process is much more simpler, and you can use minimal preclinical work and you can come much quicker to the target. I carefully work on intellectual property relating to that, and you have to turn and bend all the data and come up with innovative type of solutions. They're often related to galenic and administration routes and so forth. So that patent can be a composition, galenic preparation, or even a manufacture patent, a new manufacture process. When you look at what you are supposed to do with your product or innovation, it should be a clinically relevant improvement, or at least what you estimate, versus existing commercial products. And if you don't do that, it won't be anything worth the patients out there, of course. The effect or clinical endpoint needs to be easy to evaluate. I don't work with multiple sclerosis or some of the very long-term outcome that I work with pain. You give the patient a treatment and you get the response immediately, more or less, or similar type of endpoint out there. So, and there are some additional things that could increase the value of your project if you think of them beforehand. For example, is there a LCM strategy, sort of a strategy where you can extend the value of your product or formulation or combination? For example, through galenic formulations that are possible to upgrade. It could be novel or alternative indications. Or maybe there's an orphan drug possible. You have to investigate that. And if you get a type of indication or innovation project which satisfy more or less most of these things, I think you have a good start. So, very important, Occam's razor could be your guiding principles, for example. Simple solutions are to be preferred rather than more complex ones. Or, like Albert Einstein phrased it, everything should be made as simple as possible, but not simpler. And I think that's extremely important. Try to avoid all difficult and complicated solutions. So let's go to the pharma sector. And you can start with the preclinical basic research and so forth, and then you have 10 or 20 years down the line before you end up at the product. Or you can start at the other end and try to improve projects or products that are out there. And that is a much more simpler route. And you can even get from idea to a product that could be put on the market in a couple of years. And that's a huge difference as compared to the top-down approach. So can that be done in reality? Well, let me give you an example. We worked with this molecule, and I'm sure you know, all of you know what it is. It was discovered and identified in 1935 by the American Kendall, who got a Nobel Prize in 1950. Commercialized by a American company, life-saving many diseases, side effects, limit long-term use. And that was the problem. And still is a problem. And you all produce it right now. All of you. It's cortisone. So we started out and say, I mean, when we produce cortisone, and in the normal circumstances, we don't get the side effects that we see when we give it exogenously. Why is that? So what's very important with cortisone is that there is a diurnal production. And we said that, well, maybe we should make a tablet which satisfy high and very rapid concentrations in the morning, and then lowering and very low concentrations in the evening. And that is what we did. So this was a new formulation. This was the innovation. It was a tablet, and you can see that to your right, down there in the panel, 0.8 millimeters or 8 millimeters in diameter. And then you can see to the left the tablet. It was an immediate release coating on the outside and extended release in the core. It was a GMP produced, and it was conventional drug delivery technology. But we could patent the profile, and we can patent some of the features of this galenic preparation. So this is what we achieved. You see in the gray shaded area, that's a normal concentration range of cortisol, which normal people produce. And in the line above that panel, you can see the features of the preparation that we used. Very high concentration in the morning and very low, almost nothing at night. So that was a perfect diurnal type of preparation. So what did we achieve? Well, we achieved a new oral delivery system, simplified glucocorticoid replacement. It produced a more physiological exposure to cortisol, and it made biochemical monitoring and dose titration more robust. When we looked at the patients, we saw that that cortisol profile improved metabolic outcome. So bone markers improved, lipid profile improved, and also quality of life improved in these patients. So we sold that project, and we sold it to a large company, Pharma at the time. And this is the sale in 2011. It's the main industrial paper, Dagens Industri in Sweden. And at the same time, in the main panel, you can see the guy who was straightening up Saab at the time, which went broke. And up here in the right-hand corner, you can see a small article telling bingo for researchers. It was no bingo. It was very well planned. It was no researchers. It was people who knew product development. And when we realized the value, there was 180 million Swedish crowns, that's about $20 million, between the Saab valuation of Saab and our product to our advantage. So it was worth more than the whole of Saab. Now we realized part of that. We didn't realize all the steps that could be the milestone steps, but we realized about 280 million Swedish crowns in this project. So I want to end up with this, because our panel here are, like me, post our clinical age. We are up in the 65 plus age. And if we look at entrepreneurs in Sweden, we can see in general, when it comes to small companies, it's not the young people, it's the people like us who create projects. We have the experience, we have the networks, we have the contacts, we have the time to do it. So I think that when you mature and get up to that age, you start your own companies and you start your project. So that is what I like to say. So in summary, innovation is not a linear process. It goes back and forth and so forth. Be pragmatic when you select the project, be resilient, and plan your exit. And if you do that, you'll have a lot of fun. Thank you.
Yeah, well, that was a new aspect. A bench to bedside approach. And first I thought you should reverse that, going from bedside to bench. But actually, it turned out to be look around and see what you can do with what you already have. And I think that is a great idea. Any questions from the audience? Or any comments from you? No, well, then it's your turn.
There you go. Fourth talk. Do you have the slides? Yes, please. Versi, V-E-R-S-I. Thanks very much. Okay, so it's my job to convince you that the industry perspective is important because it's the industry that sits between the bench and the bedside, and the industry can facilitate or inhibit progress. These are my disclosures, and these are my other disclosures. I have worked for several companies, and I have had a hand in developing several products for urology. We've already discussed the translation from animals to humans, but it's critical for the industry to understand that that is robust data. The next thing we have to do is it safe? And there are standard, well-regulated toxicological studies that have to be done before you can get into humans. And we need to know if there's a preclinical signal that would make you alert to safety issues, and if there is a margin of safety for dosing. In other words, you want to have tested at least 10 times that in animals. And then finally, when you're looking at the product, it may have efficacy, but is it tolerable? Because, as has been mentioned already, it's the risk-benefit ratio that's important. And this varies by disease category. For cancer, we probably would tolerate a drug that has some side effects. For a benign disease, a lot of side effects are going to make that an unsuccessful product. So these three questions of will it work, is it safe, is it tolerable? If the projected answer to these questions is, any of these questions is no, this therapeutic will not be developed because no one's going to fund it. And I want to emphasize projected answer because at the end of the day, the people who are making these decisions are having to go by their gut as much as the science that is available to them. So that's the development side, but there's also a financial side, and as has been alluded, there are several steps that you have to go through to get from discovery of drugs over here all the way to approval over here. And as Karl-Erik mentioned, this would be sort of the T1. That is to say, first of all, you look at the molecule, look at the structural aspects of it to make you have an idea of where it might have a therapeutic impact. Then you look at receptor screening to see how it might work in the body. Then you do in vivo pharmacological models. Like we discussed, there are a few models for urology, but we will use those to see if the drug works there. And then finally, we would then decide, okay, let's go into humans, because it's a big financial commitment. But you know, the vast majority of drugs, as already been mentioned, die at this T1 stage, and that is because of lack of efficacy in these models, or there's a toxicology signal, or bioavailability. If the drug can't get to where you want it to get to, it may never actually become a medicine, and so on. But even when a drug gets into humans, into phase 1A, which is in normal healthy volunteers usually, there's only a 10% chance that it will actually become a medicine. And even when a drug passes all of this and gets down here to good phase 3 data, you still may have a 5% chance of failing to get through the regulators. And so, you know, there's a lot of risks involved here, and even after approval, there's a risk of reimbursement. If the healthcare authorities do not want to fund it, it's not going to succeed. And if doctors feel that the efficacy doesn't warrant the side effects or the cost, it's not going to work. So that will be a market failure. And yet, given all this, to take a drug to market, it costs approximately, well, more than $2.5 billion. And that's because of all the other drugs that fail. And from a company's perspective, they're looking at that investment plus also the cost of sales and marketing. And that's really the analysis that has to be made. So without a confident, risk-adjusted financial projection of a significant return on investment, because these are for-profit companies, the device or drug will not be manufactured. And so, as you can see, I beg your pardon, it's all about the Benjis. And those of you who don't follow American politics may not know what a Benji is. This is a Benji. It's a $100 note with Benjamin Franklin's face on it. Now, of course, I can't afford these high denomination notes, so I had to borrow from Karl-Erik. Promise to give it back. Okay. Then the return on investment depends on who you are, and it's very important to look at generic companies separate from branded companies. So with generic drugs can only be sold once the original patents have expired. And these companies have to do very little investment, small, inexpensive bioequivalence studies, so they have a low risk of failure, and the pricing will be determined by the market. That is, competition reduces pricings. However, you can have inefficient markets. For example, side deals with big pharma. In America, the largest healthcare provider, insurer, is Medicare. Everybody over 65 is eligible for Medicare. And yet Medicare is not allowed to negotiate with drug companies pricing, which is very, very uncapitalistic. And the reason for that is that these guys sitting over here get their campaign contributions, and so therefore they pass laws that aren't necessarily in the people's best interest. Another example is when a drug is about to go generic, the manufacturer may make a deal with one specific generic manufacturer, and therefore give them preferential treatment, and so the price is maintained high. And then there are other examples that you might have heard of. There's a man called Martin Shkreli, who his business model was to look at the landscape, find a generic drug that is made by only one company, buy it, and then raise the prices. And he raised the price of Daraprim by 56-fold overnight. And everybody screamed and shouted, including these mainly gentlemen in Congress. And in fact, it was their fault, because they should have put in laws that prevented that kind of anti-capitalistic gouging. So what's the solution? Well, we need to enforce existing laws, and we need to implement better laws. And the Justice Department of the United States is really quite unwilling to take people to trial. Instead, what they do is they come to a settlement. Large sums of money are passed from the company to the government, and who pays for that? That's the shareholders. That's you and me out of our pension funds. But the executives who did the bad stuff and got the fat bonuses, they don't go to jail, they go free. So I think we need to use the laws that we have, but we also should have decent laws that prevent anti-capitalistic approaches to drug pricing, which is why I think the drug companies today are regarded as only slightly better than tobacco companies, which is a travesty given what drug companies have done for healthcare. When it comes to branded pricing, that's a little bit more difficult, because the original manufacturer needs to recoup the investment. We've just shown you how expensive it is, and needs to provide shareholder dividends because it's a for-profit company. And before patent expiration, this is a monopoly. And if there are no other alternative therapeutics in that space, it's difficult because prices cannot be governed by the market. So what can we do? Well, we could have enforced price control. A not-for-profit model would definitely result in no innovation. And we know that because I don't think much came out of the Soviet bloc. We could have arbitrary price control. But what we've found so far is that that would kill innovation. And let me explain. I showed you the way that drug development takes place. And these days, T1 and the first-in-human studies have been relegated. Big Pharma has just stopped doing this because what they found was that the cost of R&D went up, the number of new chemical entities stayed the same, so the price per drug increased. So what they do is they let entrepreneurs and venture capitalists fund the early stuff, and then they buy it, sometimes for ridiculous prices at phase 2, phase 3. The problem with that is the person, the venture capitalist who's looking at this potential right at the beginning, if they see price controls at the end, they're not going to invest. So again, innovation goes down. So perhaps the value for money approach might be the best. And NICE in the United Kingdom has it about as good as I can think of, because what they do is they insist that there should be QALYs that are worth paying for, that is quality of life years. Because I think we'll all agree that a new medicine should improve the quality of life of people compared to the current paradigm. But also, a new medicine has to reduce the overall healthcare costs, because otherwise healthcare providers, healthcare institutions are not going to actually fund this. And I don't mean cost minimization, the cost of the drug itself. I mean the overall cost. So that, for example, if you have good efficacy and tolerability, then the patients aren't going to go back to the doctors for more treatment, more intervention, and more costs. So I'd like to leave you with some big questions. How can drugs be correctly priced without killing innovation? Is it possible for the best expensive medicines to be universally available? Is there a country that actually can afford to do that? Or should there be a two-tier system where the rich get the best expensive branded medicines, and then the rest of us have to wait until they go generic when they're cheaper? Thank you.
Thank you. Well, our time is out, but I think we have got some good information and explanation why translation into in continence medicine has been so. Your attendance here, and also I would like to panel for their contributions. Thank you very much.